We started working at the Getty Conservation Institute a number of years ago in collaboration with conservators at the Getty Museum to study their collection of French 18th century furniture that was decorated with lacquers. The lacquer that's on the furniture comes from two different general locations, Asia and Europe. So in order to understand what the lacquers were composed of, we needed a test method that could analyze both types of lacquer. So the most tricky lacquer to analyze would be the Asian lacquer, because the Asian lacquer can't be dissolved in solvent. And many of our analytical instruments in our labs require samples to be dissolved in solvent prior to being analyzed. Asian lacquers, you absolutely cannot dissolve them in anything, and so it greatly limits the test method that we could apply to studying the lacquers. So if we could develop a method that worked well with Asian lacquers, it would also work well on European lacquers then, because European lacquers are solvent-based lacquers, and so they can be easily dissolved in solvent even after many hundreds of years of aging. So the type of technique that we decided to use was a method that had been used by other researchers to study either Asian lacquer or European lacquer, but never together in the same type of sample. So the test method is called pyrolysis gas chromatography mass spectrometry. We love acronyms in the lab, so this one is mercifully abbreviated as PY-GCMS. The advantage of PY-GCMS is that you can take a small sample and there's almost no sample preparation techniques that you need to do with it, short of dropping it into the cup, sample cup, and putting it in the instrument and starting the analysis. So in terms of the analysis itself, it's quite easy. The conservator has a much harder job than I do in taking samples. It's a tedious process to take those layer by layer samples. But once those samples are taken, then I have the easy job to start with of analyzing them. So running them through the instrument's easy. The hard part comes in interpreting the results, and that's usually the case for most types of scientific tests. So the meat of what we've done at the GCI to characterize Asian lacquers and European lacquers with PY-GCMS is twofold. The first is we decided to add a reagent or a chemical to the sample before we pyrolyzed it. And we did that because we get better analytical results for the European lacquers. So that had the added benefit, as we learned later on in the project, of being able to identify materials that the Asian artists used to make the Asian lacquer panels that we hadn't anticipated at all. And it was because the method was developed to characterize both types of lacquer, European and Asian, that we were so highly successful in detecting new materials in the Asian lacquers. So the hard part is the interpretation of the data. So let me back up one step and tell you a little bit about what pyrolysis does to a material like a lacquer. So pyrolysis is just the application of high temperature to a sample. When you do that, the sample breaks down into small molecules, and those small molecules then are amenable to GCMS analysis. GCMS analysis requires the samples to be in the gas state. So solid sample goes in to the pyrolyzer. It's exposed to very high temperature, maybe 550 centigrade, and within a matter of seconds, that high temperature breaks the carbon-carbon bonds in those massive lacquer molecules, and the gases then are swept into the GCMS for separation and identification. So those molecules then are, in the best of circumstances, indicative of the original sample material that went into the cup. So you get many chemical compounds that come off of a sample after it's pyrolyzed. You might have 100 to 200 peaks in their chromatogram, and each peak is one compound. So that means we have several hundred compounds evolving that the GCMS has to separate and identify. All right, so we'll start by adding the Tmah reagent using a syringe. We'll add it to the sample cup. The sample cup's already been loaded with sample by the conservator, so our job is quite easy. We'll start by rinsing our syringe in methanol. We'll do that 4 or 5 times just to make sure the syringe is clean. Okay, that should be clean enough. So now we'll get our Tmah reagent. It's 25% in methanol, and we'll add 3 microliters right to the sample. So aim it right toward the bottom of the cup. We'll touch it off so that the droplet stays in the cup, and that's it. That's it for our sample preparation. All right, now that the reagent is in the sample cup, we're going to load the sample cup onto the holder for the pyrolyzer. And we're going to pick up the cup just like that. We'll straighten it out a bit so it's not so crooked. That's much straighter. And now it's ready to load in. So now that the cup is loaded into the handle, we'll place the handle on top of the pyrolyzer. This is the furnace. This is the area that's 550 degrees centigrade inside. So we'll unscrew this lid. We'll put it behind, and then we're going to load this right in like that. We'll tighten the ring like that. And now the sample's loaded into the pyrolyzer furnace. We waited several minutes so that the air that leaked in is purged out of the furnace. And when that's done, we're ready to pyrolyze. So then the GCMS software, the control software, it will beep at me when it's ready for me to introduce the sample. So when I hear the beep, then I push the button on top, and the sample cup drops into the furnace, and the analysis begins. All right, when you're using Tmah reagent, there's one bit of maintenance that you have to do every few samples, and that is to clean out the inside of the furnace tube with a swab in order to wipe away any residues of Tmah that might have crept out of the sample cup and touched the glass furnace tube. So we'll use a cotton swab. All you have to do once the analysis is done and the GC oven is cooled back down to room temperature, so we'll open the furnace like that, and we'll take the swab, and we're just going to run it up and down the first couple of centimeters of the furnace tube. And you see there's actually residue that the swab wiped away from the walls. So that's clean. And now we go ahead and cap it back up. And that's it for our maintenance. Now, if you're lucky enough to work in a lab that has an auto sampler on the pyrolyzer, I'll show you how to load the sample cups into that. So we'll rinse our syringe with methanol 5 times. Make sure it's clean. Okay, now that it's clean, get the reagent. And we'll add 3 microliters of our reagent. To the sample cup. And we'll put this needle right down inside, and we'll direct the reagent right on top of the sample. And we'll touch off the last droplet on the side of the cup. And now we'll go ahead and rinse the syringe again with methanol to make sure that it's all clean for the next sample. So you'll notice that the cup for the auto sampler looks a bit different than the cup for manual injection. This is the manual injection cup that you've seen. It has a hook on it, and it's a bit smaller than the autosampler cup here on the left. So this is the proper cup then for the autosampler on the pyrolyzer. So now we'll load the autosampler cup with a sample in it into the autosampler. Well, first we'll take the lid off. And then we'll use our tweezer to load the sample cup into the autosampler. And it's there. Now we'll put the lid back on. So one important aspect then of our Py-GC procedure is evaluating the complex data that comes out after the sample is pyrolyzed, the compounds that are formed are separated and then identified. And so scientists who use Py-GC have their own way of doing this data evaluation. There are many ways of doing it, but when we started our project, we wanted a comprehensive procedure that would reproducibly evaluate all the peaks. So to do that, we thought we could use some of the software that the instrument manufacturer has on the Py-GC instrument that is used for quantitative analysis. Quantitative analysis is quite a rigorous scientific endeavor where the scientist attempts to measure the concentration of different analytes in a sample. And so to do that properly, then you need to measure the amount of the material exactly. And you need to do that. You need to be able to do that over and over and over again for each and every sample and standard and reference material that you've tested. And so the procedure that we use then involves developing a calibration table of the marker compounds. So it looks like a big endless list of organic compounds, so that all the gibberish that you would expect associated with complex organic molecules and the naming and everything, you see that in the table, but you can also see when each compound comes out of the machine and is finally detected. That's called its retention time. Retention time is one of the unique characteristics of an organic compound. So then the last thing that it measures then is the size of the peak. The larger the peak in the result, the more of that compound that's in the particular sample. So it measures if a compound is present, and it tells us then how much is present. And so there are a couple of software programs that we use. The first one to actually do the quantitative analysis versus this calibration table. And the second is, we don't trust the software. You know, we're skeptical scientists. And so we want to double check to make sure that the software did its job properly. And so the second step then is to verify the retention times and the spectrum and the peak areas, then the size of the peak for each compound in the calibration table. Right now, our calibration table has nearly 400 compounds in it. So we've been busy little bees in the lab identifying these marker compounds. So now, the output of that program then is a list of all the compounds that were detected and the size of the peak for each compound. What we do then is that comes out as, say, a text document. So then we copy and paste that text document into Excel, and we developed a worksheet in Excel that sorts the many different compounds, say up to 400 of them. It sorts them by type of material, type of artist material. So all of the drying oil peaks are sorted into one group. All of the Urushi lacquer peaks are in another group, and so on and so forth for the many different artist materials that are represented in that list. So then the final step that Excel does is it graphically displays the results. and it tabulates the relative amounts of the different artist materials. So it's quite a slick and streamlined process. It sounds like a daunting task to tackle 400 compounds, but the combination of the quantitative analysis software and Excel allows most people to be able to identify all of the materials in the sample in probably 20 minutes or less. And it does it reproducibly, which is crucial for our work. I suppose to be really effective in our research, one thing that we absolutely need to have is a well-characterized set of reference materials. Without reference materials, we'd be driving our car in the dark and we would go nowhere. So the Py-GC chromatograms, the results that come out of the machine, very complicated. So it helps to know ahead of time what to expect, what to anticipate. So by getting reference materials from reputable artists, material suppliers, botanical gardens, lacquer makers, those samples then are hugely important to us and other researchers. So one of the things that we intend to do in our lacquer analysis workshop here is to discuss the idea of some kind of global reference database that other researchers would have access to, and then again getting other materials. We might find when we look at our analytical results that we haven't identified all of the marker compounds. When a compound is identified in our Py-GC system and that compound is in our database, the software puts a label over the peak. So we can tell at a glance which peaks we've anticipated, which peaks are in our database, and which peaks are brand new and unknown to us. So when we find those unknown peaks, it might be that they're coming from a material that we haven't analyzed yet. And that happened on a number of occasions throughout the research. We found exotic compounds that we hadn't expected, like wood oil from trees in Indonesia. So the wood oil was, our best guess as why wood oil was in the formulation was that it was a diluent to the lacquer to make it flow better and to alter the drying properties. The other thing we need to do then is, after we get these reference materials, we have to paint them out, as it were, prepare them like a lacquer. So we get, say, that sample of wood oil or a drying oil, we need to paint it out onto glass. Glass is a great substrate for us because it doesn't contaminate our sample. It's quite inert. So we can then paint out these reference materials onto the glass slides. We let them dry, or we cure them like an Asian lacquer would be cured. The two-step process of applying high humidity for a couple of days and then air dry for several weeks. But anyway, after the reference materials are dried, then what we would do is we would expose them to conditions that would simulate natural aging. So we have these machines in our laboratories downstairs from where we are now. They're called accelerated aging instruments. And so they're really just boxes that have strong light sources in them to simulate sunlight. What happens to a lacquer if it's exposed to sun? What happens to a lacquer if it's exposed to high and low cycles of relative humidity or temperature? The types of changes that might happen in a gallery or in an artist's studio. And so these accelerated aging instruments cycle constantly. And so in a relatively short amount of time, say a month or two, you can simulate the effects of, say, 100 years of natural aging. So now we take these samples after they've been exposed to accelerated aging, and now we start looking for, again, looking through the results to detect those compounds, see if there are any compounds that are unique to that particular material, which would make them a marker compound. And sometimes these unknown peaks in our chromatograms then actually relate to products that form with age. And so we've actually seen that happen as well. So in our collection we have 6 lacquered objects that have these panels of Asian and European lacquer. So we studied those objects with our final analytical method, our Py-GC method with that chemical reagent. And we compiled the results, and we could then tell what materials were in each piece of furniture, each lacquered panel that we tested. And each layer, and that was again a unique aspect of the work, is the work that conservators did to sample layer by layer. It gives us unique information about how the artist actually made up the lacquered piece from the very first preparation layers on through to the final finish layers. So all that information then was compiled into reports, and the reports were then integrated into a catalog of our collection that will be coming out fairly soon. So that was one mode of dissemination, but we've also knew that other people, other scientists and conservators would be interested in our test methods and how it could be used in their own research. And so, of course, we spoke about our results at symposia and conferences. Probably the most significant symposium we attended and presented our results jointly was the Crossing Borders Symposium at the Victoria and Albert Museum in 2009. And there were many lacquer researchers there in the audience. Many presenters talked about their own research in conservation, art history, and conservation science. And so our results were quite well received because many of the results were startling to them. Many people had no idea that these lacquers were as complex as they are, and their formulations were quite complex indeed. So as a result of the feedback that we got from people who attended the Crossing Borders Symposium and who have attended our other talks at conferences, we decided then to teach these analytical methods then to a wider audience of conservators and scientists. And so we again worked with our education department here to develop a workshop that we thought would be interesting to both scientists and conservators. And the unique aspect of our workshop is called Radical for Research Advances in Characterizing Asian Lacquer. I think the really unique thing about Radical is that the teaching will be done to teams of scientists and conservators. The Radical workshop then, we'll have our first one in October of 2012, and we intend to, this will be at the Getty Center in October, and we intend to repeat it at other venues then to reach a wider audience. And by doing so then, more people can find out about the test methods that have worked quite well here, but also by bringing together 10 or 20 scientists and conservators into one place for a week to study lacquers, we feel that that would actually provide the stimulus to develop a global network of lacquer researchers. And so what we're trying to do then is dangle the lacquer carrot in front of the scientists. We want to get them interested in studying lacquer because it's fascinating, it's totally understudied as a material, and we can learn quite a lot then not only about how they were made and what they were made from and trade routes, but also how they're actually aging. Our attempts to do accelerated aging in our lab, we've started with very simple lacquer formulations, maybe 1 or 2 or 3 components. But what really needs to be done is to have research to understand what the actual formulations are in the furniture. And then we can replicate those formulations onto larger panels, age them in our accelerated aging equipment, and see how they've behaved. Not only what chemicals are present, but also what they look like now, and do the aging phenomena that you see on the surface match the aging behavior of the actual objects and collections.